Hey, Aaron.
How's it
Hey, you
going?
guys.
Welcome to another episode of Ask Aaron and Dear Beth. Here we
Yeah,
go. What do we got?
we are going to look at binucleation and what makes it hard to look at in Cell Profiler, and therefore, what can we do to make it a little bit better in Cell Profiler?
Awesome sauce.
Yeah, definitely something that um, is not something you would necessarily expect to be hard, but because of some weird things about how what Cell Profiler makes in terms of assumptions,
Mhm.
can be unexpectedly a challenge.
All right. Well, let's dive right in. What is Cell Profiler expecting and how do we hack Cell Profiler? That's the
I mean, we hack Cell Profiler in lots of ways.
I know. That's probably that is much too big of
Uh,
a question for us. But like how are we going to specifically hack Cell Profiler in this exact circumstance to help us deal with these multinucleate cells?
Yeah. So Um, Cell Profiler sort of has the assumption under the hood that cells and nuclei belong together. And the reason it has that assumption is generally because when you're looking at cells, cells might be hard to figure out where one ends and the next one begins, especially if they sort of get up on each other and they're touching each other. And, here we have some cells that are actually pretty nicely behaved, but still, if we zoom in, um, sort of right in this particularly crowded region, I'm not sure I could say the exact pixel where this cell, stops and this cell starts. And you're going to run into that situation a lot with cells. Cell culture, cells are somewhat contact inhibited most of the time, but they definitely do like to sort of get pretty close to each other.
Yeah. And again,
So,
that's also a thing that's very dependent on cell type.
mhm.
But most of the time what we try to do when you're working with images of cells like this is get cells that behave nicely and don't grow on top of each other. So these are actually like pretty nicely behaved cells. There are definitely cell lines that are much harder to parse than this, where they like to start growing on top of each other, and you might be in like here, there probably is a single pixel that is the transition point from one cell to the other, that we just can't tell enough from the image ourselves to figure out what that is. And we're not even addressing the regime where there actually isn't a single pixel because cells are growing on top of each other. Like that's not even what we're talking about in this instance. Like these are nicely behaved cells for image analysis like this.
Yeah, exactly. On the other hand, nuclei tend to be much
Mhm.
nicer and much better behaved. So let's also adjust the contrast on these guys. And I'm doing this just by control clicking. Um, and you can see we've got a couple cases where things are touching, but overall, the number of cases where we wouldn't be sure exactly which pixel goes with exactly which nucleus are a lot less, and in general, things are much more well defined. So when you can get away with it, Subprofiler recommends, just sort of by its existing structure, that you start by identifying nuclei and then you build the cells around them. So here's what that looks like in practice. Um, in our object processing, we always start with identify primary objects. And what primary objects means in this case is we're building an object based on only one piece of information, the image. Uh, we're not using any other information. It'll become more clear in a second when we look at identify secondary objects and we have a second piece of information. If I go into test mode and I just leave the default settings on for now, this is probably not going to work for, oh, that actually worked pretty well.
Oh, lovely
Um,
for default settings.
yeah, it's almost like we tuned those for a reason. These aren't perfect, but they're not terrible. Um, One of the things I'm specifically looking for here as I'm tuning things are these diameter settings. And it says that in general, things are ranging between, at the 10th percentile, 16 and a half pixels across, and at the 90th, 26 and a half. And so the reason I'm looking at that is, in general, it looks like the right things are being identified. They're just being broken down wrong. And the easiest way to change that, if a subprofiler wasn't picking up on what is a nucleus and what isn't, I would need to change my thresholding settings. But if all I need to do is change how the nuclear stuff is broken down into discrete nuclei, sometimes I don't even have to do anything away from the default settings. I can just adjust the object size.
Mm.
Lovely. There we go. So pretty. Um, Now it's not doing a great job here when the objects are touching, so maybe I'll try and go down a little bit further and see if I can split the difference between 10 and 15. Otherwise, we'll, take a quick look at the advanced settings. And hey, look, 15 did the trick. Magical.
Wow.
Yeah. So now we have all of our objects, even this one, which looks like it is actually two different things. They're just very similar in the color scheme. This one says it's object 52 and that one's object 53. Awesome.
Nice. And one thing to note here is that by changing, here in our sort of easy mode, the simple mode, changing the typical diameter, like there are things happening under the hood that sort of default assumption Cell Profiler is making to help segment those apart. Like we could go to advanced settings and fine
Mhm.
tune lots of those things on our own. But
We
just
don't.
again, we don't have to like here,
Mhm.
just changing that diameter. Cell Profiler knows enough uh,
Mhm.
to figure out those objects.
Mhm. Great. So now that we have our nuclei and they're lovely and beautiful, we can add a second object. We can say, hey, cell profiler, use the cytoskel image and the nuclei and build cells around the nuclei. And again, when you have a mononucleate image, that works. And hey, look, this is beautiful. Um, we now get things that look like cells. Um, They're a little on the sort of underside, so, we might change our thresholding method to try to pick up a few more things. But again, the goal here is not to have things perfect. The goal here is just to get some things that are plausible cells. Okay, that didn't do a whole heck of a lot. I could tune it more. I don't really need to. You get the idea. We had nuclei, we made them into cells. Great. I'm going to actually just change the default scaling here as we're looking at stuff, which I can find in the settings. And Mac, it's called preferences. And that's just going to make it so that things generally look prettier and it's a little easier to follow along with what we're doing here. Now, how does that now look? That looks worse. All right. Um, so mhm. 80 was okay. We'll go with 99, just because that seems like a fun number.
And one thing to point out, this isn't doing anything about changing the underlying data. This is just how it's being shown to us
Exactly.
here,
Yeah.
as we're looking at these
We can now see the cell boundaries a little bit more easily and just in
yeah,
general get a better sense of what's going on.
yeah,
Yes. This is just a display visualization. And yes, now we can see these much more easily too.
yeah.
Great.
And here
So,
perhaps, oh, sorry. Just
uh-huh,
want to, also point out that like depending on your task, you may be spending more or less time in your, ID secondary to get perfectly the very edges of your cells.
mhm,
Here we're just, not spending the time fine-tuning because our task right now is just to explain to you about multinucleate cells. So we
mhm,
just need to find edges that sort of make
mhm,
enough sense that we can go through this demonstration. If you were to be doing quantitative image analysis, on the cell objects, perhaps you would be iterating more at this point.
for sure. Um, Now let's get a sense of what this is going to look like in the case of multinucleation. So I'm going to tell Cell Profiler to let me pick an image set. And now I'm going to go down to the ones from our positive control image and get some cells from there. Okay.
Whoa,
So,
look at that.
so Cell Profiler has, in some cases, it has messed up and sort of put two nuclei into one. In some cases, the nuclei are so big, there are so many of them that this has now turned pink, which means it says this thing is too big. I think it's all one thing, and I think it's too big of a thing. It's a thing that's bigger than what you told me you want. I'm throwing it away. Um, and in some cases, it's doing kind of the right thing, though those cases are pretty rare, to be honest. So here we have what I would count as five, and we have one, one, and three. So not great, but potentially even more not great is when we look at our cell calling. So the fact that we sometimes have cases where what is clearly two nuclei have been stuck into one actually helps us get the right cell mask because Cell Profiler is still assuming that nuclei and cells are one to one. But in this case, where it actually did do a good job of splitting some nuclei off, now we're in a terrible position in that this cell is now broken into chunks, which is definitely not what we wanted.
Yeah,
Sadly.
that does not, that does not look like the cells we want. I think
Mhm.
cell chunks, anytime it's just like
Mhm.
not a descriptor that we generally want in our biology.
So I really have kind of three choices here. Um, One of them is um, I can try to force Cell Profiler to make all of the nuclei that touch be one thing. So all of these nuclei would be one thing. This cell wouldn't get broken up. But in cases like this big chunk here, where there are multiple nuclei that we're touching that are actually part of multiple different cells, those things are now going to be stuck together. I'm also not going to be able to count for multinucleation because multinucleation, the nuclei are being detected sort of intentionally incorrectly. But if cell boundaries are important, that might be the best thing to do.
Yeah, so one thing to really consider at the beginning of building your pipeline is, what is your output that you're looking for? What is your downstream task? What are you trying to measure? Like in this situation, you know, maybe you don't actually care about any measurements of the nuclei. Maybe what you're really concerned about is something happening in the cell. And, so all you really need is the cell boundaries. And then and then we don't actually care to identify the individual nuclei parts. But maybe that's actually really important if your downstream task is to measure the number of nuclei, or something related to a measurement in the nuclei themselves. And so strategy of any workflow is definitely very dependent on the output that you want.
Absolutely. Which is why we want, even though we are professional bioimage analysts, we're good at doing bioimage analysis. We want people who are just biologists who want to answer bioimage analysis questions to understand how to do it. Because you understand ultimately if it's more important to know more about the cells or more about the nuclei. And so you are the best person to design your image analysis workflow. We're
Absolutely.
always happy to help, but your input is not optional. It is essential to us designing the right workflow, even if we are the ones designing it. Uh, so I said that was one option. The second option is to, instead of trying to force Cell Profiler further into the direction of sort of stick stuff together, we can try to force Cell Profiler more into the direction of split stuff apart. It's going to get our nuclei correct, but it's going to mean that if we keep using this ID primary and ID secondary, that we end up with cell chunks. Which again, maybe is fine because if what you want to say is are the multinucleate cells different phenotypically than the other ones, they sure are. They sure are different if this cell is only a piece of a cell as opposed to a whole cell. So if what you care about is just detecting phenotypic difference, that might be the right way to go, is to just have cell chunks. The other third option is to abandon this ID primary, ID secondary sort of workflow entirely and just use completely separate identifications for our nuclei and our cells. Then we can try to get the best nuclear segmentation we can. We can try to get the best cell segmentation we can, but if the cells are really clumpy, that might not be very good cell segmentation. There's a reason we do this in the first place, and it's that cell segmentation is typically kind of hard. Um, in the interest of what we really hope you get here more are the concepts. The cell segmentation here actually, now that you understand how it works, doesn't really matter. Um, if I wanted to force the nuclei that are all touching to be all the same object, that's one very easy setting in identify primary objects. That's just method to distinguish clumped objects. None. Do not distinguish clumped objects. If nuclei are touching, just call them all the same thing. Um, I'm now going to have to adjust my sizing amounts
Yeah.
because now these really big things are being told that um, they're too big and Cell Profiler doesn't want to count them anymore. But I could do that. That would be one way to go.
That's so easy. And a good thing to notice here, like we don't actually think that there are nuclei that are 500 pixels, in diameter. That was just like throw out a large number, because we have
Mhm.
it will filter the objects outside of the diameter range. So if we just pick a really large number, then we don't have to worry about it using it to filter. But because we have this sort of in advanced settings mode, especially,
Mhm.
it's not using that to approximate anything else or affect anything else.
The other things we could do are a little bit harder to walk through in a short thing. We will have a future episode. Aaron, remind me to have a future episode
All right.
on declumping when things are really crowded. But we can certainly do things that would push Cell Profiler towards forcing the nuclei to be smaller and more separate. Um, and there's pre-processing tricks we could go to actually push this even further, but we've now at least gone further than we were before in that we definitely have some sort of smaller nuclear chunks. This one is a little bit better. This one is a little bit better. But this is hard. I mean, especially if we zoom in on some of these ones that aren't doing a good job, even that. Like, I think those are two different. I think there are four nuclei here, one, two, three, four, but even I'm not totally sure.
Yeah. I mean, one could, yeah, yeah. I don't know. Or it could be three, right? Like maybe it's, one horizontally,
Yeah,
two at an A. I don't know. That's tricky.
I think that's a ridge. I think there's four, but it's hard to say.
It's very hard
Um,
to.
but
And
if you are thinking of, sorry.
I was just going to say, I think that's a good uh, a reminder about sort of basic principles of image analysis, right? Like here, we are two expert biologists, and we are having a very hard time saying where is the line on this nucleus. The computer can only do what we tell it to do, right? So, when we get into this, this computational regime of thinking, it still relies on you still rely on expert biologist input to this task. Like it will only do what we tell it. We need to be able to tell it to tune it to the regime that we think matches biology best. And so sometimes you can only get so much, you can only get what you can get out of the image. Like maybe it's just not possible to tell. And then uh you know you can't do better than a that you can't do better because you have no sort of ground truth or expert biologist vision to compare it to.
Yep. And if we really cared, we could probably do things like we could look at the other channels and try to get a sense, like I was saying before, that I thought this clump was five, and then when I looked at the cytoskeleton, I thought it was six. So there are things we could try to do. Um, but for now, I think we shouldn't because this is the case where we really want to turn to deep learning. Now, we don't always want to turn to deep learning for everything because deep learning, it's harder to understand when it will fail. But there are really good deep learning models for segmentation now, and our very, very favorite is a tool called CellPose. If you haven't tried CellPose yet, you absolutely should. It is wonderful, made by the folks at Genelia. Great free open source tool. Um, and here we're running it in Cell Profiler using a tool called Docker, which allows it to run in its own little container, which means I didn't have to do any difficult installation. To install CellPose on my machine, instead of having to go to my terminal and try and do things, which many of us do not want to do anything in our terminal, I opened up the Docker desktop program and I searched for CellPose, and then I looked for the one that was said Cell Profiler run CellPose. That was all I did. And then I looked at I added the Run Cell Post Cell Profiler plugin. You can check out, I believe it's Weisbart et al. 2023 to learn more about Cell Profiler plugins and how to use those.
Yeah, and maybe that's a good future episode idea there too. Maybe we should
Mhm.
go, deep dive into the plugins ecosystem. So another one to add to the list.
Um, but so here I'm telling Cell Profiler that I'm going to be using CellPose with Docker, which means I need Docker open and on, which I have. That I want it to detect nuclei, and I'm going to tell it that I think my nuclei are about 15 pixels across because that worked pretty well before. I'm not going to try and tune any of the other parameters right now. There's lots of other parameters in CellPose. Like any cell profiler module, there are help functions next to each one of these things, and you can get the help about what is going on with each parameter by checking the help. Always the most important button in Cell Profiler. Um, why are you unhappy? I think you aren't unhappy. I think you're just pretending.
I think it's just pretending. I think that's
Yeah.
an outstanding issue that is not affecting functionality at the moment, but affecting what it's t it's a little
Mhm.
cranky, but it does actually work.
Yeah. So CellPose is really good at figuring out when stuff is clumped, how to actually split it apart. And so often when we are doing multinucleation workflows these days, and by these days I mean in the past 5 years since CellPose was first proposed, we that is the case where we don't try to do things with classical cell profiler anymore, but we actually leverage these deep learning modules because the fact that they're a little harder to install and that they're a little bit harder to predict when they will fail is outweighed by the fact that they just work better at these tasks. So we're not never deep learning people, and we're not always deep learning people. We're deep learning when you weigh the factors and the factors balance. This is unfortunately pretty slow in Docker in Cell Profiler 4. When we put out Cell Profiler 5, this will be much faster, at least the second time. On the first time, it's always going to be a tiny bit slow because it has to actually make a whole system for running CellPose. It's the downside of it installing really quickly is that it is not actually going to run super quickly. Okay, there we go. As soon as I navigated away, it finished. Um, so here that input image looks terrible because of the normalization stuff. So let's just put that back to normalized. But you can see with literally zero parameter tuning other than saying how many, like what is the approximate size of the objects, CellPost has done a way better job than our initial things did, and certainly better than I think even if Aaron and I spent a bunch of time doing it, I think Cellpose would probably do better in the long run. Like
Ah.
this super big chunk that we were never able to split apart. It hasn't perfectly split apart. I still think there's a couple things that are stuck together, but it is way, way better. And there's more stuff we could do to tune things. So if you have a multinucleation workflow and things are super clumpy and it's really important for you to be able to detect the binucleation, using CellPose and using CellPose even sometimes twice wants to be able to identify the nuclei and wants to be able to identify the cells um, is actually the way to go because when it is important, when things are likely to be clumpy and weird looking, then CellPose is probably going to do the better job. And again, this will probably take about a minute to run. Um,
So what are the things you just tuned in this module, Beth, as you were chatting?
Yeah, no, that's a great question. I tuned which cell pose model to run. I changed it from nuclei before to cyto here, which is their cytoskeleton marker or their sort of whole cell model. Because we now want to find the cells, I switched which image to use. I told it to use the cytoskel image instead of the DNA image. And CellPose has actually been trained to assume that cells have nuclei, and that cells have. It might be tuned to have cells one nuclei, so that might actually hurt what I just did. It might actually make it worse by turning the supply nuclei image as well on,
Hmm.
or maybe not. I'm not actually sure. And that's the sort of question where you could try and read the documentation, or you could just look and see what happens. And I always like to look and see what happens. Nothing against documentation. I read and write lots of documentation, but sometimes you just want to see in this exact circumstance what's going to happen.
Yeah.
And with deep learning, it can be really hard to tell, which is why we don't always go to deep learning first. But I did tell it that it could have a nuclear image as well, and I also changed the expected diameter. We said nuclei were about 15 pixels across, so I said let cells be about 25 pixels across, and that was vibes based. Um, Put this now back to normalized. I don't know why cell pose. I think cell pose is normalizing the image itself. So it hasn't detected our super big cell because it's super big, and it has actually broken things to where the nuclei are. I'm going to try running this one more time, and I'm going to not give it the nuclear image, and we're going to see if it'll do a better job. I think it's still not going to pick up this cell because the cell is just so big. There's stuff we could play with, but that's another episode. But let's see if we don't give it a nuclear image, if some of these things it gets a little better, like this cluster.
When we give it the cell diameter, like, do you think
Mhm.
there'd be a situation where bumping up the cell diameter in this setting, a
Mhm.
bit, because I think 25 seems to be on the smaller end of things. Maybe that will also help it. Interestingly,
Mhm.
here, right? Like, so in our identify secondary module, our identify primary and identify secondary, when we're putting sort of the object diameters in those examples, we were using them as hard cutoffs, saying, don't give me an object that is outside of this range that we put here in CellPose, this run CellPose module. This is different. Like, this isn't a hard cutoff
Mhm.
for object diameter, where again,
Yeah.
we're not,
There is also a hard cutoff. It's this minimum size parameter. But yes, the expected object parameter isn't. It's a guiding fact. Um, we want to be careful about setting that too big, though, because we want to make sure that this pipeline also works for the ones where we still have little individual cells. So we don't want to go crazy. We don't want to make it too big.
yeah.
But yes, we can see if those changes help.
And that's a good principle, right? For when you are building a pipeline, you always want to make sure that you are testing it on, if
Mhm.
you have positive and negative controls, great. Make sure you're testing it on both of those. As Beth mentioned, like any parameter that we're setting up here to work on these cells, we need to make sure it's also going to work on our negative control population where the cells look a lot different. And so having a broad enough test during your pipeline development is essential.
And so it doesn't look like actually those changes made a whole lot of difference, which is not the answer I would have expected. But again, deep learning can be a little bit hard to predict when things go well and when they don't. There's a lot of other cell pose parameters we could test and we could tune, and there's a lot of other cell profiler ones we could. But what we really wanted to do here is just sort of illustrate to you what things you might try in terms of thinking through the problem. Do we want to sort of prioritize nuclear correct? Do we want to prioritize cell boundary correct? Or do we want to sort of go to this deep learning situation, which is a little harder to predict, but may work better? And all of those are valid pathways, potentially, depending on the biology you want to measure. And all of those could then be further tuned to get the exact best answer for your data. So I don't think the tuning will teach you as much as the principles, because the tuning will be specific to the data you have.
Absolutely.
Any closing thoughts, Aaron?
No, no. I mean, I think this is, it's a fun challenge. And again, I think an interesting thought exercise in remembering that, like, there are many very valid approaches that will give you very different measurements in the end. And so paying attention to what is my task and what do I need to, what am I prioritizing in accuracy, is definitely something to keep in mind that has, I mean, I think for any workflow, but it has a major impact on this particular example.
Thanks for sticking around and hanging out with us. It was great to see you and hope to see you next time.
Bye.